Hello everyone, welcome to GetMiClass. So let's solve this question here. It says the virus enters into macrophages where the RNA genome of the virus replicates to form viral DNA with the help of the enzyme dash. Option A, reverse transcriptase. Option B, DNA polymerase. Option C, RNA polymerase. Option D, helicase, DNA helicase. So let us understand the question. The question says the virus enters into macrophage where the RNA genome of the virus replicates to form viral DNA with the help of the enzyme dash. Okay, so what are they asking? They're asking about the viruses which actually enter into macrophage, and the RNA is the genome of the virus which will get replicated to form viral DNA. Okay, so with the help of which enzyme? That is the question. And option A, they have given reverse transcriptase. Okay, so what is reverse transcriptase? Yes, this is the enzyme which helps in the formation of, or if you say helps in the synthesis of DNA using RNA as the template. Using RNA as the template. Okay, as the template. Now, if you see, so reverse transcriptase is the one which is actually associated with the main virus, that is RNA virus, especially in this case, we're talking about macrophage. That means we are talking about the HIV virus. Okay, so here the HIV virus genome is RNA, and that is associated with the enzyme which is called as the reverse transcriptase. So usually transcription is from, like, DNA acts as a template for the formation of mRNA. But here it is the opposite of it. That is, RNA is forming the template. For example, this is the RNA, and using RNA as the template, DNA has been formed. What is this? DNA. So this process, it happens in a reverse fashion, right? Where usually it is from DNA to RNA, but here it is RNA to DNA. That is why we call it as reverse transcription. And the enzyme involved here is reverse transcriptase. So this is the correct enzyme. Option B says DNA polymerase. So DNA polymerase helps in synthesis of the DNA. Synthesis of DNA, and this is useful in the what? In the replication of DNA. Okay, it happens, or it is useful in replication of DNA, or you can say even repair mechanism. Okay, but this cannot convert, or this cannot code for DNA using RNA as a template. This is DNA dependent. Okay, hence this is ruled out. Option C is RNA polymerase. RNA polymerase is also an enzyme which is again DNA dependent. DNA dependent in the sense it needs DNA as a template. Dependent. Okay, DNA as a template. It needs DNA to be used as a template, and that is what will convert or will code for mRNA using RNA as the DNA as the template. For example, this is the DNA. Okay, RNA will be formed or synthesized by the RNA polymerase. Okay, this is RNA, and hence we call this and this is the enzyme, for example, RNA polymerase. RNA polymerase. Now this RNA polymerase uses DNA as the template and not RNA as the template. Hence we call it as DNA dependent. Hence it is ruled out. Now option D says helicase. Helicase is used in separation of the DNA strand. Of the DNA, double-stranded DNA, you would say. Double-stranded DNA. Okay. Suppose you have double-stranded DNA like this. Okay, and these are of course bonded by hydrogen bonds, right? Between these nitrogen bases. Okay, so and this helicase will combine, it will bind to the DNA and will break this hydrogen bond, and that's why it will separate the two strands. But this is not involved in the replication process. That means it doesn't form the DNA or synthesize the DNA. Hence it is ruled out. So what is the answer? The answer is option A. Let us conclude. So option A, that is reverse transcriptase, is the enzyme which will code for DNA using RNA as a template, and that happens in the macrophages which have been infected by RNA viruses. Okay, I hope you understood the explanation. Thank you, everyone.